Good morning, everyone. Welcome to Gramene webinar. This is Sushma Naithani from Oregon State University. Today, I will talk to you about analysis and visualization of plant-gene expression atlas data using Gramene, Genome Browser, and Plant Reactome. So this is the Gramene homepage. As you know, you can browse various tools and various functionalities of Gramene database. I will not go into detail of these today. However, you can go to the search, and I would like to introduce you to Grameen new search page. Here is Grameen new search page, and here, as usual, you can find about the latest news that we have in Grameen or updates. You can also enter from here to various portals of Grameen, such as Genome, such as Plant Reactome, Blast Tool, and so on. So here also you have a connection for Plant Gene Expression Atlas. So if you click here on Plant Gene Expression Atlas, it will take you to Gene Expression Atlas homepage. Here you can browse plant experiments. Currently, Plant Expression Atlas contains about 674 experiments from 15 plant species. If you go here on this page, you find two types of data. The first type of the data is baseline experiments, which means that these are the experiments conducted under normal conditions from different plant species. And here you find expression data for various tissues. And so various tissues and tissue-specific expression data is here. The other type of the data here comes from differential experiments, where one or more tissue parts are treated under certain conditions, and you can find the gene expression data related to these particular experiments. First, I would like to talk to you about baseline expression data. Baseline expression data in EBI-Atlas is all from RNA-seq experiments. Currently, it is available from 10 plant species, and soon it will be available from 12 species. The expression level of gene products were recorded under normal conditions for various species. So, for example, here we choose Oryza sativa japonica to look into baseline experiments. So, as you see here, we have about 4 different studies from which the data is available here. So I'm choosing this tissue baseline experiment data from Davidson et al. So you can click on individual experiments. So if I click here, I can open up these data, and here what you see in this column gene IDs of all the genes for which expression was recorded. And this side are various tissues such as seed, anther, carpel, seed, and so on. Again here, dark blue represents high expression, and gray is almost no expression, and light blue means less expression. So what you could do further, you can select any genes from this particular very long list. I have chopped that list here for convenience. So you can click on individual genes, and what you come up with, you get expression data for that particular gene across all tissues that for which the data is available. And this time you don't only see the expression data from Davidson et al., but you see all those data that is available for rice from 4 studies represented here. So these are from 4 studies, and as you see, you can see expression data for various tissues. This particular widget here can allow you to see into the vegetative tissues versus the reproductive tissues here. So you can change it anytime, but you can see the expression data. Similar data you can also access. We have integrated this data into Gramene gene page. So if you go to Gramene gene page and you click on gene expression, you can pull out the expression data for given gene that has been recorded in all 4 experiments that were available for rice across various tissue samples. Again, here in Gramene gene page, you can see the transcript table for that particular gene. So if you click here on transcript table, what you see that there are two transcripts or two isoforms here, and you can also further go to the UniProt and look for also the protein sequence and so on. Again, if you want to see this data in Atlas, you can click here. So we have integrated gene expression data between Atlas and Gramene in very seamless way. Again, if you go to Atlas data, you can have data from a single experiment. And if you want to visualize this data on genome browser, what you need to do, you need to select a gene here and one column here. And then what you could do, you could click on either on genome browser that I will show you, or you can also click on ensemble genome browser. So in both sides, when you click, it will open genome browser, and here in genome browser, then you can have the gene you have selected in the center, and you can have the expression data. So you can't upload the data for more than one sample or one treatment at a time in genome browser, but you can do one by one, and your browser will remember it, and you can see all those data at the same time or simultaneously in the genome browser after a while. So here first I chose expression data from Enter, and the second time I chose expression in the leaf. And as you can see that although I had selected this particular gene, but the expression data is also loaded for various other genes. So you don't need to select all genes one by one. You can just select one gene, and it will load the experiment. It will load the data for all other genes that are available, for which the expression data is available in that particular treatment. So this is how you can express. You can jump also to other region in the genome browser and see see the genes in which you are interested. So now I'll tell you a little bit about how gene expression data is linked to plant reactome. So this is our plant reactome page. You can come here by clicking on Gramene pathways, or you can come directly at plantreactome.gramene.org. So simply, if you click here on browse pathway, it will open this pathway browser. Just for a little orientation, the left-hand side of this pathway browser has this list of the pathways that are available in plant reactome, and you can open by clicking on plus sign, the further details or the reactions that are listed under a pathway. On this top right hand side, you have the pathway diagram viewer, and here you see a pathway that is depicting a pathway in a eukaryotic plant cell having different compartments. So for example, this particular pathway, jasmonic acid signaling, is taking place in two compartments, cytosol and nucleoplasm. And you can also see various different types of reactions, including metabolic reactions, signaling reactions, and so on. So again here, in the plant reactome, this bottom panel shows a lot of information that is associated with a given pathway. So one of that information is about expression. So if you click on expression, you see the same baseline gene expression profile that is available on EBI or Gramene gene page. You can also see that in context of the pathways. So here, all the genes that were mapped to the pathways or identified or associated with a given pathway, you can see their expression pattern across various different types of the tissues for which data in EBI is available. Again, from here, if you like to go, you can go to EBI and go jump to the experiments and again go back to genome browser. So we try to integrate these features so users can jump from one platform to another platform and do some useful analysis. Now I'll talk to you a little bit about differential expression data. So here I just wanted to say that it is available for 15 plant species, but As you see, Oryza, sativa, indica, and japonica both were separately listed, but so I just wanted to bring that point. So differential gene expression data is available from both RNA-seq and microarray experiments. They are available from 15 plant species, and expression level of these genes products were recorded under various treatments or conditions that are described in respective experiments. So again, you can select any treatment or any species from here, and then you can look into individual experiments. So I'm selecting here again Oryza sativa. And for Oryza sativa, we have various experiments available here. So this is a longer list. This is a short description here. And again, I'm choosing this particular experiment, the first one, and it is a transcription profiling by array of wild type and drought and salt tolerance mutant Japanese rice vine. And if I click on this, again, I see that expression pattern. And what I see here is a relative expression. So it is a relative expression of genes in the mutant rice vine that is drought tolerant and salt tolerant. And here what you see the fold expression. So you see the log 2 fold change. And so the red is upregulation and blue is downregulation. So again, there are different ways how you can visualize this expression data on Genome Browser or on Pantry Ecom. So simply for Genome Browser, you can select any of the gene from this list, and then you can select this. There is only one column here that is depicting upregulation or downregulation, the ratio. And once you choose these two columns, then again you can click on Gramene Genome Browser, open here, and you will open the Genome Browser page. And what you see here is mapping of the gene expression profile on Genome Browser. And you can jump from one chromosome to another chromosome. And so here I will show you how you can visualize differential gene expression data on Pantry Ecom. So you can actually use our analysis tool that is used for analysis of omics data similarly here. And in Pantry Ecom homepage, if you click on Analyze Data, it will open the window for uploading omics data. So in order to upload data on Pantry Ecom, what you need to do, you have to first go to EBI-Atlas page and select this particular experiment or your favorite experiment. And here, then you can download the data, and you can download also the result from here. So you can download here. So once you have downloaded this particular data in a text format, then you can basically have two columns. So one can be the gene ID, and then second can be your ratio. So this has a lot of other information. You don't need that. So what you could do, you can rearrange gene ID, and then you can have the expression values that next to it that you want to visualize. So you can reformat your text file here, and then you come to Pantry Ecom, and here you have this analysis tools for omic data analysis. This is the icon for this. So you can come to this page by clicking on this icon from any other page in Pantry Ecom. So here you have option to upload this file simply by browsing and then uploading, or for convenience, you can also just copy the data from two columns of text file, and you can copy and paste here that I have done here. And then you hit Analyze. So once you do that, you will see a window depending upon where you are. So if you choose here a higher level category that I have done, metabolism and regulation, you can see a picture of few related pathways that are related, that are listed here. And you see some color coding here where you see mapping of genes that show differential expression here. So here, for example, secondary metabolic biosynthesis, and you can go further and then browse individual pathways. You can go to hormone signaling and then see which are the hormone signaling pathways that are upregulated or downregulated. So in the bottom panel, then you can have list of the pathways and you can browse. And if you click on a given pathway, you will see the diagram for that. So what I'm doing here, I'm going to click on the GA12 biosynthesis pathway. And once I see GA12 biosynthesis pathway, I see that one gene in this particular pathway that is catalyzing 3 reactions of gibberellin biosynthesis is highlighted. So this is upregulated here and it is mapped here. So you can have that information and have and analyze your gene expression data in context of pathways, and you can go from one pathway to another, or you could choose to see the expression of related pathways. Also, the pathway mappings and pathway diagrams you can download by clicking here in the results and also in the mapping. So here in this particular example that I took from RICE, EBI-Atlas, we had only one column or one treatment that showed the ratio. There are also times when you can have omic data analysis and you have more than one sample, and you can similarly upload all of those as a text file in Plant Reactome and conduct that analysis. So here is another example that I would like to show you. So this is data from other source, and as you see, we have sample 1, 2, 3, 4, 5, and 6, and that shows the gene expression. And here what you have, not the gene ID, but you have the uniprot ID. So you can have gene IDs, also you can have uniprot IDs, and you can have the expression data. Similarly, for compounds, you can have compound ID and then you can have metabolic data. So Plant Reactome allows analysis of more than one type of omics data. So here again, if you copy and paste here and say analyze, this is what you get. So it is relatively a larger data set, and you see various pathways that show blue color here in this particular sample. And then if you go backward or forward, you can see various samples and find the analysis of and mappings of those genes that show up or down regulation in given samples. So this is what you can do. Again, here you can download those results or pathway mappings. And here in the list also you can see in which pathway how many genes were identified that showed upregulation or downregulation. From here you can go to an individual pathway. So here again I'm going to this pathway here, and this pathway we have found that these two enzymes show differential expression, and interestingly this box is not completely blue, but it is partly gray. And so let me tell you what it means. So here if I click on this one box, then I see listing of 2 genes. So because there are in plants there are a lot of gene duplications, and sometimes there are very big gene families, and all of those genes that are homologous, they are mapped to the enzyme, same enzyme. But it is not necessary that they share expression profile. So some of those can be shut off in a particular tissue, and others they show upregulation or downregulation. So as a plant scientist, we know that gene duplication is very very common in plant genomes, and identification of the candidate genes that are responsible for certain trait or certain processes within a given developmental phase in any plant is a challenge. So this analysis actually allows you to differentiate between the expression profile of homologous genes and mapping to the pathways, and allows you to postulate a hypothesis that can be useful for that can be useful for designing a more focused experiments and cut down on some unnecessary parallel experiments. So this is very useful feature that I would like to share with you. Again, you can go from one sample to another sample by clicking forward or backward here. You can also download results here, and that's it. You can find our webinars, previous webinars on Plant Reactome, genomes, blast and all in the Gramene YouTube channel. And we will be happy to host any webinar that you suggest us or you need help with. So please let us know. You can contact us by clicking on any of the Gramene All-Plan page. You can write your message here and send it to us. We are usually good in responding. And finally, I would like to thank NSF for funding Grameen project and its collaborators. I would especially like to thank Robert Pritzak for developing plant gene expression atlas in EBI. I would also like to thank all my collaborators in Grameen project, people at OSU in Pankaj Jaiswal's lab, people in Doreen Beer's lab at Cold Spring Harbor Laboratory, folks in EBI and folks in Reactome project at OICR and NYU, and Crispin Taylor and his team at ASPB. Thank you very much to you for listening, and we welcome any questions or any suggestions on webinar topics. Thank you very much. Have a good day.